starting out with your warm-up on 90. What is the end result of mitosis? What do you get? 2 identical daughter cells, right? Are they haploid or diploid? Diploid. What is the purpose? Why do I do mitosis? To produce what? So I'm producing them. Why do I have to produce them? What does it sound like? Mitosis. What does it sound like? Toast. So it's for making toast cells, growth. Growth, repair, and asexual repro. Does it make identical or different? Identical. Describe what occurs in the steps. What happens in prophase? What do I do? Huh? Condense. That's our prep phase. What happens in M? Middle. Line up in the middle. What happens in A? Apart. I pull apart. What happens in T? I form. How many? 2. I go back into those 2 nuclei, right? What must I do before I do mitosis? What's the big part? I have to. Huh? What happens in interphase? That's very, very important. DNA replication. I must replicate my DNA. Gotta replicate that DNA before I'm able to even do mitosis. If I don't replicate my DNA, it's gonna say, hey, can't do it. Okay. 91. Human body starts with blank chromosomes and ends with blank. How many chromosomes do you start with? 46. And how many do I end with? 46. Good. And meiosis, my gametes have how many? 46, 46. 23. Good. Which of these is a reduction division? Meiosis. Which of these makes identical cells? Mitosis. And which of these makes different cells? Meiosis. Good. Remember, meiosis makes me. That is sexual reproduction. Mito. That's growth. And that was it for your warm-up. So today we are starting on page 57. 57. With unit 5. So we are almost to the end. You have 57. 57. Almost to the end. We're only going to make it through. How many times I see you? Unit 6 by the time I see you, because the last day I see you will be a cram session. So unit 5, molecular genetics. My central dogma. What is this molecule? DNA. What is this molecule? RNA. And what is this molecule? What is this? Huh? It is a peptide. What's another word for peptide? Protein. Protein, right? How do I go from DNA to RNA? What's the process called? Transcription. Thank you. How do I go from RNA to protein? Huh? Translation. Where does transcription occur? What organelle? Nucleus. And where does translation occur? I hope you know that, Louise. Where does translation occur? Ribosome. Ribosome. So we started Unit 5 with this central dogma. This was your main idea of biology, the fact that we can take DNA and convert it to protein. You started your unit with DNA. So your DNA is made up of a sugar, a phosphate, and a base. What is the sugar in DNA? Deoxyribose. It makes up the sides. Typically, it is a 5-carbon sugar, so it's going to look like a little pentagon. Phosphate is a phosphate. Nothing special about it. This is page 57. It is also on the side, and it looks like a little circle. So that's your sides of the ladder. And then your bases make up the rungs. What are your 4 bases? G, A, T, and C. A, T, C, G. What's the shape? What do we call the shape? Huh? What's the shape, the spiral shape called? Double helix. Remember, it looks kind of like this. Like a windmill. And then what are your base pairs? A pairs with T and C pairs with. Good. What are the bonds between them called? Hydrogen bonds. Okay. The pyrimidines and the purines? Purines are 3. No. I think it's uracil, adenine. No. Because it's like A and C. It's like 2 of them have 3 bonds. I don't know. What about RNA? Ribose. It's still inside. It's still 5-carbon sugar, so it looks like a pentagon. Phosphate, still a phosphate. Still the side. So lovely little circle. And then you get that uracil. Uracil pops up here. So uracil replaces thymine. What's the shape called? Single helix. Who bonds who? A pairs with Q, and C pairs with is still an H bond. Who discovered DNA? Rosalind Franklin. And who's your other guy? Watson and Crick. Rosalind took that X-ray photo. You might see it named by name. It's called Photo 51. Watson and Crick were the 3D model. Both showed it's a double helix. That illustrates our nature of science, the fact that science is constantly evolving, scientists are constantly collaborating with each other. Last step is protein synthesis, how that works. So DNA replication, you told me it occurs where? In the where? DNA? Nucleus. And what do? Replicate DNA, right? A copy. Want to make sure that each cell has equal amounts for my cell cycle. What's transcription? DNA to RNA. We're still in the nucleus because DNA can't leave the nucleus. And then translation is what? mRNA to proteins. And that happens at the ribosome. So what is mRNA? Messenger, good. My messenger RNA. So it's going to carry information from DNA. Think of it as you went to go look up something and you're going to write it down on a post-it. The mRNA would be your post-it. You looked in the textbook, you said, hey, I need to write this. I wrote it down, that's my post-it. Are you ready to flip? Not yet, okay. Okay, on the back. 58. Almost done. So your other molecule is tRNA. What does tRNA do? What does it stand for? No translate. Close. Transfer. Transfer. This is the one that's going to bring those amino acids to the ribosomes. It's the one that's going to go searching for those amino acids after it reads that codon. Can you see that? What is a ribosome? Organelle. This is also rRNA, ribosomal RNA. What's a codon? How many letters is a codon? 3. Where is it on the mRNA or the tRNA? mRNA. Triplet based mRNA. Yes. Triplet. Remember, we use the codon for our codon chart. And so what's the anticodon? Nope. It's the opposite of the codon. So who's it on? On the tRNA. Does that match mRNA? And then your last part, why do I care about these codons? Why do I care? Why did I send a whole unit on it? It's because proteins determine what you look like. Going into your next unit of Mendelian genetics. What you look like. That's the point. That's why we care about it. And then the last one is the end of the unit was about synthetic biology, about how we manipulate our genes in order to do specific stuff. So you start with the polymerase chain reaction, otherwise known as PCR. PCR was a cool technique we use just to amplify our DNA sequences. It's to make more of them present. We're basically doing replication on steroids. This is how you can take a little bit of DNA and make it a lot of DNA. You'll use this with like crime scene evidence. You can use it in disease analysis as well. Let's see, seed analysis. That's your forensics. and then disease diagnosis. PCR is typically coupled with gel electrophoresis, which is where we can take that amplified DNA and then separate it by fragments. So you might see when you're analyzing, we've done it where it's like, there's your code, and then it's like, who's the father? And it's like, here's your babies. And it'll be like, who's the dad or whatever. That's gel electrophoresis. This is how you get your DNA fingerprint. CRISPR is where you get into your genetic modification. CRISPR is where we can edit genes. So this is where we can add different genes in. We can say, hey, I don't like that gene, let's take it out. So we do that for treating genetic diseases. This is how they make insulin for people who are diabetic. This is disorders. So they actually grow insulin in culture, in bacteria. Human insulin help us treat diabetes. And we also see in agriculture, you'll hear things called GMOs. They used it to edit genes. This plays right part with recombinant DNA. Recombinant is basically what you get at the end of CRISPR, where you're combining DNA from different organisms. So you took your DNA, you took a specific spot you want, and you ended up getting something that has that embedded. So whether that's how we make vaccines, GMOs, human insulin, and bacteria. Your last two on cloning, huh? Yes. Your last two on cloning. Cloning is whenever we create an exact copy of an organism. Obviously, a lot of ethical implications with cloning. Is it actually good for us to use it? Big one with cloning to know was Dolly. Dolly the sheep is where I made an exact copy of that somatic cell. Huh? Where are you going, Crystal? Oh, yes. Exact copy of somatic cell. You did not use a sperm or egg. They think this would be really good for our endangered species, any type of regenerative medicine. They're trying to regrow your organs. And the last one was the Human Genome Project that happened in 2002, where they sequenced the entire human genome. They said the gene for eye color is on this one. The gene that makes you male is on this one. Where your genes are for which one, so we can understand genetic disorders, evolution, prevent and treat diseases. They actually can find out now if you have breast cancer in your genes, if you are genetically susceptible to breast cancer. They can find out if you're genetically obsessed to being diabetic. Are you genetically obsessed to being blank? Because they can sequence your entire genomes. What you're doing today. So on station 1 is all about DNA. First task is labeling. And then evaluating the picture. So you're going to label this. So knowing that the whole part of it is called a nucleotide. So I've labeled that part. The whole thing is a double helix. Labeling that part. A, C, T, and G, make sure you match them. You're going to do just a phosphate, just a sugar, and label those as well. When it says evaluate the effectiveness, you're going to say what you can see from this picture. What's the only thing this picture can tell you? What about DNA? The fact that it is a ladder, and the fact that your base pairs are A, C, T, and G, right? Sugar and base are the rungs. Ooh, sugar and phosphate. A pairs with T, C with G. Task 2 is a fill in the blank. 3D model, Watson Crick. Task 3 is a little bit about genetic engineering. What are benefits of synthetic biology? So you have a little kind of case study there at the bottom. Start right here, I'll go back here. On station 2, you're going to do DNA versus RNA transcription translation. You're going to compare DNA versus RNA you're going to use the words in the word bank. So basically, you're going to take these words, place them in the bank wherever they go. Task 2, describe the steps of transcription using the following DNA strand. You're basically going to describe what's happening, the fact that RNA copies DNA, then it heads and leaves the nucleus. Task 3, same thing with translation. mRNA goes to the ribosome. The tRNA reads codons at a time, matching the anticodon, bringing over the amino acid using my strand. And then task 4 is to label. So using these words, you're putting them and labeling them on that picture. And then station 3 is all multiple choice. Then you have your lovely little quiz on page 62. So you have 3 stations, 3 pages, and then a quiz at the end. This one?